All right, it's noon, and I guess we should start. So before I start, I would tell folks who are attending, who have already been attending this seminar series for the past, crazy, it's like 8 months now almost, which is the Connect Science series here. And as you know, all the talks are recorded and available on the website shown on the screen. So you can go visit that, and couple of issues, just logistics. All questions should be directed in the Q&A tab at the bottom of your screen, and I will ask as many as possible to our eminent speaker today. And if they don't get answered, we will try to get back she will get the questions and might get back to you if they're interesting enough. All right, so let me introduce Professor Carol Privis. It's such an honor to have her here. She's a professor at Columbia, was the chair of biological sciences from 2001 to 2004, and has been a professor since 1987. I mean, I literally can show you a page full of accolades, so I'm not going to go through that. I'll just do a couple. One is that she just got into the Royal Society this year, and she's been in the Academy of Sciences, Arts and Sciences here since 2000. But I was looking at her CV, and the first paper in Carol's CV is a Nature paper with her as first author, 1969, Effects of Cerebral Stimulation on the Biosynthesis in Vitro of Nucleotides and RNA in Brain. It's brilliant. I think this is so relevant today, and this is a Nature paper with her as the only first author and her PhD advisor, I believe, J.H. Quaster, in 1969. So you can imagine how this started, and 250 papers later, I mean, she's really a pioneer in P53 amongst other things, and that's how I came across her work. And it's just amazing that every other year she comes up with something which we, in a molecule which is probably more well studied in cancer biology than anything. But Dr. Privis's papers are just a different dimension and makes you sit back and think. So I am truly honored to have you here. I wish it was in person, but we'll have to make do. So I will let her give her talk now. And
well, thank you. That was really embarrassingly flattering. I will say that my first paper, which was a letter to nature, was literally a letter to nature, because in those days you said, "Dear Sir," and then you said, "I wish to report," and you'd give the numbers as part of the narrative. It was pretty strange. All right, that's not what I'll be talking about today, but I'll be talking about, let me see if I can figure out how to move this slide. This is going to be, how do I move the slides? How do I advance the slides? Do I just?
You should just be able to click.
Oh, I did it.
Yep, there you go.
Okay, thank you. So thank you again. And obviously I'm not working on cerebral stimulation of brain slices, but I am working on P53, and I've been doing so for decades, along with a very large number of other people. It's a very, very active and very supportive and collegial field. And the interesting thing about P53 is the more you study it, the less you know, which is a good thing and a bad thing. So we do know, and you can see this on the slide here, that P53 itself is extensively and very carefully regulated by two proteins, MDM2, a transcriptional target of P53, and its partner in crime, MDMX. And they keep P53 levels very low unless P53 is needed, and it's a stress response factor, and I'm not going to get into this today, but as a result of many different ways of impinging on this circuit, P53 levels rise. And when P53 levels rise, you get a wide variety of cellular outcomes, many of which are consistent with tumor suppression. I'm not going to go into all the reasons why we know P53 is a tumor suppressor, both in animals and in humans, but we do know a lot about how this works, even though we still don't understand fully what exactly it needs to do. And one hypothesis is that it needs to do all of these things in different contexts. And so we know that P53, whoops, I'm sorry, I'm not sure how to, no, this just changes the slide. How do I do the animation? I think I use my mouse. Okay, so we know that P53 does good things. It suppresses from tumor. It's been shown to play a role in fertility, but too much P53 or deregulated P53 has been associated with bad outcomes such as neurodegeneration, premature aging. And one of the many things that P53 does, and it was first shown to regulate cell cycle and then cell death and then DNA repair. One of the things that it's been shown to do also is play a significant role in metabolism. And most of the people who study metabolism in P53 are looking at glycolysis and related processes, the TCA cycle, pentose phosphate shunt, amino acid metabolism. The area that we've gotten into actually deals with lipids. And lipids are perhaps one of the more complicated set of molecules. I was at a lipid meeting for the first time, maybe also the last time, last year. There's over 800,000 different lipids, so it's very intimidating, but they do fall into different classes. And the first kind of lipid I'm going to talk about actually is cholesterol. But before I do that, I want to point out that just as P53 is incredibly central and important, so are its regulators, MDM2 and MDMX. They have the ability to bind to P53. They have a lot of other homologous regions. They have a ring domains at their C termini, but of these two proteins, only MDM2 has an active ring domain that plays a role in E3 ligase, and MDM2 also has signals that get it into and out of the nucleus. There are also sequences that determine how these two proteins form a heterodimer. And we've actually used mutants of these that contribute to the results we've talked about. So we know that MDM2 regulates P53 by E3 ligase, by proteasome-mediated degradation, by directly inhibiting its function to serve as a transcriptional activator. And also it can actually monoubiquitinate it and evict it from the nucleus. There's not a big reason for me to tell you this because I'm going to be talking in the second part of this seminar on how MDM2 and MDMX function in the absence of P53. And this is a growing field now with increasing lines of evidence that these two proteins together and possibly separately play enormously interesting roles in cells on their own that have nothing to do with P53. So the first part of the talk is going to deal with P53, and the second part is going to deal with what MDM2 and MDMX are doing without P53. One of the reasons why P53 is so interesting to cancer biologists is the way it's mutated in cancer. And it turns out that although there are many examples where P53 protein is lost due to frame shifts or nonsense mutations, there's an unusually high number of missense mutations that occur in P53. The clustering is shown in this slide. Of these, there are a known set of hotspots mutations that occur with very high frequency, and very often these mutant forms of protein are present at very high levels in cells. And it's believed, although somewhat controversial, that these mutant P53 proteins have gained new functions that are pro-oncogenic. And the first part of the talk is going to deal with our evidence that was published many years ago by a wonderful MD PhD student who's now actually at the he's working with Tyler Jacks. And it basically involved the ability to downregulate the mutant P53 in a cell line called 468 breast cancer cells. And this caused a morphological reversion in those cells to more normal kind of little acinar-like structures. And Will did a lot of experiments to support this observation. But along the way, he also acquired gene expression data. And it turned out that a pathway that was very significantly affected by the levels of mutant P53 in cells was the mevalonic acid pathway. This is the pathway by which cells synthesize cholesterol, isoprenoids, coenzyme Q, and it plays roles in glycosylation as well. It's a very important pathway in cells. And in fact, inhibitors have been developed most commonly and most pharmacologically important in the terms of how often they're used and are the statins, but additionally inhibitors of farnesylation and geranylation have also been very useful experimentally in science. So just to go quickly through this pathway involves at least 18 or so different genes that produce products, protein products that catalyze various reactions. Many of them are enzymes. And as a result of which, this is what leads to cholesterol and all these other proteins. But this is an example of extraordinary coordination of gene expression. And the master regulator of gene expression is SREBP2. It's one of a very small number of proteins. That stands for SRE binding protein 2, and SRE is a sterile response element in the promoters of these different genes. And Will developed the hypothesis that mutant P53 was somehow facilitating or increasing the activity of SREBP2 to activate transcription of these many genes, at least in the cells that he was studying. I'm not going to get into all the data that he produced to support this, other than to point out that we had a collaboration in those days, which was kind of pre-TCGA, or at least we weren't using TCGA, with a woman, a very good scientist in Norway called Annalise Borisson-Dale, who had multiple breast cancer data sets. And her data indicated a very interesting correlation between having a high number or rate or percentage of P53 mutations, a high expression of mevalonic pathway genes. And in fact, there was even survival data implying that the patients with these high levels of these genes had worse survival than the patients with low levels of these genes. So, I mean, I'm collapsing a lot of work into just a few slides, but I'm leading up to a more recent set of experiments that were done that we just published in 2019. And it basically started with the fact that we know in the P53 field that there's a very odd reciprocal relationship between wild type and mutant P53 in its ability to in their respective ability to regulate genes, such that many of the genes that are actually known to be good transcriptional targets of wild type P53 appear to be downregulated in cells with mutant P53. And by contrast, cells that are known to be upregulated in cells that have mutant P53 are very often repressed or very at low levels in cells with wild type P53. And so the question was pretty obvious, having shown that mutant P53 correlates with higher levels of mevalonic genes in at least one and perhaps other breast cancer cell lines. We asked the question whether perhaps wild type P53 might actively repress the expression of these genes in cancer cells, the presence. And so these experiments were done, spearheaded essentially by Sung-Won Moon, who is a former postdoc in the lab. He's now got a position in a company in San Diego. And he did, I think, really impressive work. He had wonderful collaborations here at Columbia, including Will Fried-Pastor while he was still in the lab, Kaushik Reganoth, who did a lot of computational work, and David Tong, a current MD PhD student in the lab, who is a real whiz with generating cell lines that either lack or express mutant P53 endogenously using CRISPR-based editing. But most importantly, in terms of acquiring more physiological relevance, was a collaboration with Scott Lowe, and particularly two people in his lab, Chun-Hao Wang, a former graduate student, and Shauna Houlihan, a current postdoc in his lab. Scott and ourselves and other P53 researchers in the New York area share a program project grant, and we have frequent or not frequent enough lab meetings or multi-lab meetings. And it was at one of these lab meetings that when Sung-Won presented his work, he initiated a collaboration with Scott's lab. So what did Sung-Won found? He found that when you turn on P53, the levels of several of these mevalonic pathway genes goes down. These are derived from a liver cancer cell line. This is particularly relevant to the mouse data I'm going to be showing later. Their analysis was in hepatocellular carcinoma, but also HCT cells. These are a colon cancer cell line that were many, many years ago, pre-CRISPR, engineered to have lost expression of the wild type P53, thereby becoming P53 null. These cells are, to a first approximation, isogenic. And when you turn on P53 with a P53 namely nutlin, which separates P53 from MDM2, you get significantly reduced levels of many of these genes at the RNA level, to a much lesser extent than in the P53 null cells. Furthermore, if you look at metabolites, which is the end product of this pathway, several of these metabolites are expressed at significantly higher levels in the null cells than in the wild type cells. So moving on, with the idea, let me first say that this general anti-correlation, you'd say, holds with many different cell lines. This is the cancer cell line encyclopedia. There's nearly 500 cell lines with gene expression data in them. And when Chen Hao analyzed it, it turned out quite strikingly that wild type P53 harboring cells all had, on average, much lower levels of many of these enzymes than did mutant or null cells with respect to P53. So that was encouraging. So the next challenge was to figure out how this repression is working. And what Sungwon did was first do a chromatin immunoprecipitation, and he found, when he looked at SREBP2, which, as I told you, is a master regulator, the mature form of SREBP2 is a DNA binding protein, and it binds to the sterile response elements in the promoters of the mevalonic pathway gene. And he found, and this is just one, at the HMG-CR gene at the SRE, he found significantly more SREBP2 binding than in cells that had wild type P53. So this led to the idea that just as mutant P53 may cooperate with SREBP2, perhaps wild type P53 is somehow inhibiting the ability of SREBP2 to act as a transcription factor. So this is now a somewhat old, but nevertheless, I think, valuable diagram of how these sterile response element binding proteins are working. And there's actually 3 of them, SREBP1, 1C, and 2. They're all master regulators. They all regulate lipid enzyme transcription. And SREBP2 in particular is the one that regulates the cholesterol biosynthesis pathway. So they're all held as inactive precursors in the ER by a protein called SCAP. They are then, upon sterile depletion, if you have too low cholesterol, these precursors are chaperoned to the Golgi, where they're subjected to 2 successive proteolytic cleavages. This releases the active portion, which has the basic helix motif that binds to the sterile response element and activates transcription of these genes. Just as sterile depletion activates this pathway, having higher levels of cholesterol in cells will repress this pathway. So anyways, Sungwon tested the idea that perhaps what's going on is that maybe the maturation of this protein is being affected by the presence of P53. And that is what's happening. There is a higher, this is the precursor form, the inactive form, and this is the mature form on a gel. And in the P53 null cells, assuming you can activate this pathway, and you can do that by using a specific serum called delipidated fetal bovine serum, this activates the pathway, and you get a significantly higher amount of this mature form in cells that lack P53. In fact, you can see this not only in the HCT cells, but in this liver cancer cell line. If you induce P53, you get significantly less mature SREBP2. And in a cell line that we've engineered to have inducible P53, again, turn on P53, have less mature SREBP2. So that's an interesting result. Now, how does that work? And that became a real bear of a problem for Sung-Won. He must have spent at least a year trying to figure out how this works, knocking down all the known genes, for example, in the maturation pathway, didn't see anything. Read widely, which is what I recommend students do and postdocs do and everybody do, because sometimes you'll see something that gives you a clue. And he finally got a clue. This is the question he posed. And he finally got a clue from reading a very interesting paper, which was a paper in JBC. So that led him to a protein called ABCA1. Many of you may have heard of these. These are these ABC transporters, and at least ABCA1 and these other ABC proteins are known to play roles in export of cholesterol. So why would a protein that exports cholesterol actually play a role in SREBB2 maturation? So I'll explain that in a minute, but here's why we focused on ABCA1. And here's the paper that I just referred to. It was a group from Japan who played a role, who reported that ABCA1 plays a role, actually an unexpected role, not only in export of cholesterol, but also in retrograde transport of cholesterol back in from the plasma membrane to the ER in mice, and can thereby basically inhibit this maturation process. But also, we had some clues that maybe ABCA1 is connected to P53. Sung-Won looked carefully at data from Joaquin Espinoza, a wonderful scientist in Colorado, who did something called GROW-seq, where you can look at direct transcription targets of a protein, looking at the RNA as it's being elongated. And he identified among the many targets, many known targets of P53, but some new ones as well. And one of them was ABCA1. And then we looked at our own RNA-seq that we had done with Nutland-seq treatment, and we found that we too had identified ABCA1. Also, ABCA1 has significant cancer relevance, particularly with liver cancer. It's the one that's most significantly down or reduced in hepatocellular carcinomas from TCGA compared to these other ABC genes. And what we discovered was that ABCA1 levels inversely correlate with mature SREBB2. And this experiment was done in P53 null cells so that we could leave out any confounding, let's say, effects of P53. So these are P53 cells from which we removed P53, and I'll be coming back to these types of cells in the next section of this talk. But you can also induce ABCA1 with an agonist, which has this name, and you can see you can induce the protein, but you can also, of course, knock down ABCA1 with siRNA. And what Sung-Won found was that there was a direct correlation between the presence of ABCA1 and reduced mature SREBB2, both in the cells that were controlled. And then when he knocked down ABCA1, he had much higher levels of this SREBB2. So can we further connect ABCA1 to P53? And the answer is yes. If we induce P53 with Nutland in these SKF1 cells, we get a significant increase in ABCA1 RNA and protein. And in these cells where we can induce either wild type P53, we see a lot of ABCA1 being induced. But if you use a P53 that has a crippled activation domain, you don't see any ABCA1. So, and also finally, we could find P53 associated by chromatin immunoprecipitate at 2 potential P53 binding sites in the ABCA1 promoter. So we think we have pretty good evidence that ABCA1 is a real P53 target, and that's how it's working in these human cells. But the validation in the mouse cell lines, I think, really gave some more physiological relevance to these findings. And so many years ago, Scott Lowe, and among the many different, I think, real wizardry he displays in setting up mouse, various interesting mouse models akin to wonderful scientists such as Tyler Jacks and others, had developed technology where you could knock down mutant P53. It's the TET-off, so you could put back P53 by reinducing P53 with basically with doxycycline. And it turned out that when in those experiments, I should have cited the paper here, P53 tumors regress. But cell lines from the immortalized liver progenitor cell lines were developed. You can turn on P53 in those cells. And when you do that, you can get a gene expression analysis, which was done a long time ago. And what Chun Hao did, because that was his work, went back and re-examined the data and found that when you reactivate P53, there's a big reduction in expression of many of these different mevalonic pathway genes. And not surprisingly, there's an increase in ABCA1 gene expression. But he went on to do some experiments that were more relevant as a direct collaboration. And this is an experiment where you can inject into mouse liver via hydrodynamic tail injection constructs expressing an oncogene such as MYC and a tumor suppressor such as P53 or another tumor suppressor axin that plays a role in the Wnt signaling pathway. And in each case, you get within a few weeks, you get liver tumors. So this is a very fast way of doing, I think, an important cancer biology experiment. So the experiment then was to say, well, if you get a reduction in these pathways, in the mevalonic pathway as a function of wild type P53, does that have any significance to liver cancer? And so what Chun Hao did was include in this, oh, let me tell you something else too. Isolating tumors from these animals and making cell lines, you could see much more of the SREBP2 mature form in those tumors that lacked P53 than those tumors that lacked axin. So as I said, the question then is, could he really tie in a more, I think, functional way the ability of P53 to repress this, this pathway? And the experiment, therefore, was to knock down genes in the mevalonic pathway. He used SQLE, LSS, and HMGCR. These are all genes that are playing a very important role in the mevalonic pathway, validated that these shRNAs work. And so now the mice got both knocking down P53 with CRISPR, guide RNA, MYC, and then shRNAs in constructs that also had GFP, so you could follow tumor formation with these shRNAs. And so the experiment was really pretty striking, the results. Knocking down any of these genes, SQLE, LSS, or HMG, showed a significant reduction in tumors in these animals compared to just the control. Whereas if you did the same experiment with axin, okay, as another kind of tumor suppressor, knocking down these genes had no impact. So that linked, I think, in a more direct way, the ability of P53 to repress the mevalonic pathway with the ability, at least in this cancer model, of normal liver cells to turn into cancer cells. So that's not particularly useful if you're thinking of therapy. So the experiment then is to use some drug that shuts down the mevalonic pathway, of which there are abundant types. There are all these statins and others. And so what Chun Hao did was pre-treat the mice with atorvastatin and then follow tumor formation. And he found, in fact, that pre-treating the mice with atorvastatin led to both smaller tumors and fewer tumors when he treated the cells with atorvastatin, where there was no impact virtually of doing the same thing with the mice that had received the axin as the tumor suppressor in that model. So again, that linked loss of P53 to increase mevalonic pathway gene expression to increase liver cancer. I'm not going to show the data that Shauna then did, but she actually looked very, very early on in this experimental protocol of tail vein injections, just a few days after injection, and she already saw a big reduction in mevalonic gene expression, mevalonic pathway gene expression, consistent with the idea that this is a very early event in the development of a liver cancer, at least in this experimental model. So then finally, Shauna did another experiment, which is to ask, well, if ABCA1 is a P53 target that is critical in possibly in protecting essentially from overexpression of these genes, the mevalonic pathway genes, and maybe ABCA1 itself might have tumor suppressive ability. So she did the experiment where she used guide RNA. She actually tried several of them, and they all worked to different extents, some more than this and some others. And she showed that very clearly there was a significantly increased number of tumors in the mice that lacked expression of ABCA1. Now this is knocking down P53, and obviously you can see a big difference here. And I think this just attests to the idea that, once again, P53 is a major tumor suppressor because it does more than one thing. And any one P53 target is not going to, at this point, phenocopy to any extent what P53 itself can do. But it definitely brings, I think, supports the hypothesis that regulation of the mevalonic pathway is a critical component of P53's tumor suppression, at least in the context of liver cancer. And just want to say as well is that when you looked in these ABCA1 null tumors, you saw significantly more in cell lines derived from them. You saw significantly more of the mature form and actually of SRBP2 itself in these cells, but there was more mature SREBP2. Okay, so basically to summarize this part of the talk, and I'm looking at my timing here, I'm okay, I think. To look at this part of the talk, I should keep my phone off so I can see my time. Okay, yeah, so basically, once again, it seems to all center on SREBP2. Mutant P53 cooperates with it to stimulate this pathway in certain settings and in certain cell types, whereas wild type P53 mediated by ABCA1 is actually inhibiting this pathway or keeping it at levels low because when it's unrestrained, it's going to contribute to tumor suppression. And so in either case, we think that there's interesting new directions to follow to look at context. You can see when this is happening, whether ABCA1 is the only target of P53 in this context, and what aspect of the mevalonic acid pathway is actually contributing to tumorigenesis. It does lead to a hypothesis that we believe is worth pursuing. And the idea is that in many cases, especially mevalonic pathway gene expression, I apologize, this slide is not quite sort of centered the way it should be. We believe that wild type P53 represses it. If you lose P53, that's loss of function. You're going to have increased, as I've shown you. But we believe as well that under certain conditions, you're going to have missense mutant P53 further stimulating this pathway. Looking at the TCGA, we see that some genes clearly fit this hypothesis, such as IDI1. Others do less, for example, MVK, there's not much difference. And in other cases, the difference is subtle, but we think it's probably real. Okay, so now I'm going to switch gears. And I'm completely switching gears because I'm not only not talking about P53, I'm talking about instead of cell death. And I'm talking about a kind of cell death that I'm sure many of you have heard of called ferroptosis. It's kind of the new kid on the block in terms of cell death types. I mean, I won't say it was discovered uniquely by Brent Stockwell at Columbia, but he certainly is a major figure in this particular field, and it's a real field. I mean, they have ferroptosis meetings where a lot of people come to them. So I guess a lot of people work on it. The work that I'm going to describe was spearheaded by a wonderful graduate student, Divya Venkatesh. She's now left. She just left a few months ago, and she's doing her postdoc at the Sloan Kettering, working on immune checkpoint therapy. And she was helped by a great undergrad at Columbia named Nick O'Brien, who's now doing a postdoc at the NIH. But this was a real collaboration, just like my previous project was a real collaboration with another laboratory. Brent Stockwell was the co-mentor of Divya, and she really did work really between our two labs. And we collaborated, as we did before, I didn't point this out, with a group at UC Denver to do metabolomics with us, but also Farishte Zankarimi did a lot of metabolomics as well. So let me tell you a bit about what she discovered. Just want to say there's multiple forms of cell death regulated by P53, apoptosis, necroptosis, pyroptosis, and ferroptosis. We know this. Let me tell you a bit about ferroptosis first. So ferroptosis means iron-dependent cell death, and it was discovered, at least a lot of the components of this pathway were discovered in Brent Stockwell's lab. And he's a chemical biologist who isolates and characterizes small molecules that affect cellular processes. And the first one he ever discovered was arrestin, showed that it can kill cells, and he then figured out how arrestin is killing the cells. Turns out that arrestin and other derivatives like it block system XC, which is also known as SLC7A11, which transports cysteine into cells, which allows the levels which is needed for the synthesis of glutathione, which is an antioxidant. And so ferroptosis essentially is a process by which cells die because of lipid peroxidation. And these are lipid hydroperoxides that are toxic to cells. This process requires iron. It requires a protein, an enzyme called GPX4, that basically converts these hydroperoxides to a less toxic form of lipids. And what Brent has discovered were many of the inhibitors that inhibit the different stages of these. There's two inhibitors of GPX4. There's actually a protein called, there's a protein, it's not a protein, it's a small molecule, Pheno2, which actually stimulates ferroptosis by increasing the levels of these lipid hydroperoxides in cells. So these are the drugs that, that Brent has developed, characterized, and worked on, and they are the ones that Divya used. And ferrostatin is a very critical anti-ferroptosis drug. It's a good way of knowing whether the cell death you're looking at, if it's inhibited by ferrostatin, there's a good chance it's ferroptosis. So the first person who linked ferroptosis to P53 was Wei Gu, a colleague at Columbia, who showed P53 can inhibit SLC7A11. He also discovered SAT1 as another target of P53 that increases lipid peroxides. Maureen Murphy did beautiful work linking GLS2 to ferroptosis and P53 as well. But it's complicated, as this slide says. There's reports that have shown that, for example, P53 can suppress ferroptosis under certain conditions, and also it can block DPP4 activity. So it's a complicated situation. And Divya waded in. The first thing she did was to basically survey a bunch of cell lines, maybe 10 or 12 of them, found that some of them were very easily killed by arrestin and others weren't. She also found that there was no correlation with P53 status. She also took cells. We have multiple cell lines now from which David Tong in our lab has removed P53 expression using CRISPR knockout strategies, and found that, in fact, these cells, I'm sorry, I've gone too fast. What have I done here? Yes, she showed that these cells, in this case, these are HT1080 cells, which are very commonly used by people studying ferroptosis. They're a rhabdomyosarcoma cell line. She showed that these cells can die, but as you can see, these knockouts, let me see if I got this right. Yeah, these knockout cells need more arrestin than the wild type cells. So P53 is contributing to ferroptosis. And this is just showing that you lose that effect when you treat the cells with ferrostatin. But she also so the idea was at first, maybe she'll study P53 in ferroptosis. And so she she decided, well, if they normally can induce ferroptosis, maybe if she can make P53 better by inhibiting its ability to interact with MDM2, you'll get more ferroptosis. And she got the exact opposite result. So using either nutlin, and I can explain why she got this if anybody's interested, or a very important compound called MEL23, which was actually isolated in Brent's lab earlier, and we collaborated with him on this. MEL23 inhibits the activity of the MDM2, MDMX, basically heterocomplex, from serving as an E3 ligase. And we did experiments in vitro, and he did experiments in vivo to show this. So this was a surprising result. And it also made us think, maybe MDM2 is playing a different kind of role in this. Maybe it's actually facilitating ferroptosis on its own. So that's what I'm going to tell you it does. And we used several different ways of showing this. And we also showed that MDM2 and MDMX are working together on this. We used siRNAs, we used MEL23, we used an MDMX inhibitor as well. And it inhibits transcription of MDMX. I don't know how, but it does work. And I'm sure that every one of these things have off-target effects. But our idea is that if you use different ways of inhibiting a process by different approaches, if they in common do the same thing, at least we can basically focus on the common thing they do rather than the off-target things they do. So here's the experiment she did. Here's what happens. This is a phase contrast. These are what these cells look like. And we also used SKHEP1 cells. When they're treated with vehicle, they look like this. They look really sick and miserable and they die. And we have many ways of measuring cell death, which I'll show you in a moment. But if you treat the cells with either the MDMX inhibitor or MEL23 or siRNAs, which I'm not showing you, you don't get very much cell death. And this is just a measure of this. This is a measure of viability. It's called an ATP measurement assay. We've used other ones as well. Big drop in viability, big rescue when you ablate either MDM2 or MDMX. She also showed that you get an increase in ROS, lipid ROS specifically. This is BODIPI analysis. This is just showing what Erastin does alone and how either ferrostatin, which inhibits ferroptosis, or these inhibitors of MDM2 bring it back to baseline. So then she tested every single kind of inhibitor that Brent had developed in his lab and found that they all block all classes of these fins. This is RSLC that inhibits GPX4, FIN56, which inhibits GPX4, and FeNO2, which causes lipid peroxidation. All of these things were basically more or less reversed, or certainly at least semi-rescued by these inhibitors. And so because of this, it occurred to her that it's something to do with lipids. And so I'm not going to get into the logic, but it's pretty clear cut. And so basically what they then did was lipidomics, both in-house and also with the University of UC Denver. And in many, many cases, first of all, it was a huge amount of data to look at. And it wasn't your typical gene expression data where you have a lot of different algorithms to figure stuff out. Just looking at them by hand carefully, it looked like very, very often there was an increase in lipid abundance when either you inhibited either MDM2 or MDMX. So this led to other ideas. And in fact, a work from Scott Dixon and his colleagues showed that you can, if you just add more of certain kinds of monounsaturated fatty acids, you would actually produce resistance to ferroptosis. So this led to a search for how this is working. So basically, MDM2 and MDMX are regulating lipid synthesis in some way, okay? And by inhibiting them, you get more lipids. And so how is this working? So this is just to show you that we didn't at the time know how it was working. All we knew is that you antagonize MDM2 and MDMX, get altered lipid mechanism, and you get ferroptosis. And this altered lipid metabolism is somehow basically promoting ferroptosis. So if you inhibit them, you get less ferroptosis. All right. So this is just a very short list of things that we considered that Divya tested. No changes in cell cycle that could impact on this. Nothing seemed to change in its localization. Many known modulators of ferroptosis did not seem to be involved in this. So what was going on? So a paper that was published a few years ago, and I'm blocking the names of the authors, and I apologize, basically linked MDM2 to another. So I'm now bringing up another master transcriptional regulator, and this is a transcriptional regulator of genes that are involved in lipid synthesis, and that's PPAR alpha. So the PPAR alpha, gamma, and delta basically control many different kinds of lipid metabolism. And I do apologize for this very busy slide where you can't read anything. I was looking yesterday if I could find something better, but none of them seemed worth it. So I'm gonna have to make one myself, I guess. Anyways, PPAR alpha transcriptionally regulates numerous genes involved in lipid biosynthesis. And so we ended up focusing on that. And I think our data do support the idea that one of the things that MDM2 working with MDMX does is actually dampen or repress PPAR alpha. So the first thing you want to do is see whether PPAR alpha plays a role in ferroptosis. And it turns out that if you stimulate PPAR alpha with an agonist, you get less ferroptosis caused by Erastin. And conversely, if you inhibit using an antagonist, PPAR alpha, you get your sensitivity to a ferroptosis inducer is significantly reduced. So how does one connect this then to MDM2 and MDMX? So this is an experiment where what Divya did was treat cells with Erastin, and then with either the ferrostatin, just to show she's looking at ferroptosis, or the MDM2 and MDMX inhibitors, MDMX and MDM2 inhibitors. And just like I showed you before, they can largely reverse the amount of cell death caused by Erastin. However, if you antagonize PPAR alpha, you don't see this. And what this means is that MDM2 and MDMX should be upstream rather than downstream of PPAR alpha, which is consistent with this earlier report that MDM2 somehow inhibits it. Mechanistically, nobody knows how this works, including us. But to further show that they are upstream of PPAR alpha, Divya looked with Nick at different transcriptional targets of PPAR alpha and found that they were greatly increased when you treated the cells with either of the two MDM2 and MDMX inhibitors. And I should point out, this doesn't show up as a change in PPAR alpha activity. So it's not like MDM2 and MDMX are degrading it. They're doing something different. And I think that's one of the challenges we have in the future. I'm finally going to finish up with pretty close to on schedule, some experiments that extend this a little further. And these were prompted by two papers that were published in Nature late last year. In other words, just a little over a year ago, from two labs. I guess Scott knew about this work, and Brent Stockwell knew about this work in advance. Some of it had been published in meetings, and he's good colleagues with these scientists. And it's all about a protein called FSP1, which actually plays a role in regulating coenzyme Q, which is actually a byproduct of the mevalonic acid pathway. And FSP1 basically maintains high levels of reduced coenzyme Q in cells. And it's reduced coenzyme Q that has the most antioxidant activities in cells. And it's coenzyme Q, according to their model, plays a very important role in basically suppressing ferroptosis. So we decided to look at FSP1 because of, we just decided to look at it. Well, it's too complicated to explain. But the most important thing is that we already knew from our lipidomics analysis that coenzyme Q is actually dramatically regulated by MDM2 and MDMX. So if you inhibit them, you get a much higher level of reduced coenzyme Q in cells. And a much higher level of reduced coenzyme Q is going to be suppressing ferroptosis. And so FSP1 plays a role in maintaining the levels of reduced coenzyme Q. So the title's a bit misleading. It's basically saying that because of this data, we can conclude that MDM2 and MDMX maintain levels of oxidized coenzyme Q, which is not helping to protect from ferroptosis. All right, so this is the data that they generated with FSP1. It's quite dramatic, actually. Both MEL23 and the MDMX inhibitors significantly increase the levels of FSP1 in cells. And these are just two different cell lines. However, if you treat the cells with an antagonist of PPAR alpha, you see much less of an increase. I just want to warn you that the order of samples in these gels was different. Here, the controls here, and over here, MDMX and MDM2 inhibitors, and ditto for over here as well. So basically, inhibition of this complex increases the levels of FSP protein, and this in turn is affected by PPAR alpha activity. So this is what we have. Basically, we now believe that by blocking MDM2 and MDMX, and the MELs are important. There's two different MEL compounds. They both inhibit the complex. We believe it's the complex that's at least partly important. Not in other words, it's not MDM2 and MDMX working solely separately. They need, at least in part, to work together. And so therefore, they probably have a target. That target is not PPAR alpha. Somehow they're affecting its activity in some way we don't understand, trying to do some experiments to figure that out. Either through PPAR alpha or directly, they can also regulate the levels of FSP1. In other words, what MDM2 and MDMX would do would be keep its levels lower. And as a result of this, they would have less of the antioxidant response and you would get more ferroptosis. So that's where we are with this. It was published in Genes of Development just a few months ago. There are many questions to pursue with this, not the least of which is how are MDM2 and MDMX regulating PPAR alpha? Are there other activities in this process? Are there other targets of MDM2 and MDMX that play a role? But I think it's an interesting observation in another way. And that is that if you tie in MDM2 and MDMX to ferroptosis, which I believe we've done, and it's P53 independent, the implication might be that, that MDM2 and MDMX in certain contexts could actually be tumor suppressive rather than tumor promoting. And that's because in certain contexts, the process of ferroptosis may be tumor suppressive rather than maybe tumor suppressive. On the other hand, we also know that ferroptosis plays a role very likely in certain pathologies such as neurodegenerative diseases, certain kidney diseases. Regarding kidney, there's a little literature that actually implicates MDM2 in kidney function and dysfunction. So, again, it's interesting to study these two genes in their own right. And again, just thinking, more generally, context is everything. How you actually study a protein and the ways in which you study it may give you really different answers. And that's why cancer research is so interesting. And I'm going to finish up this experiment with this seminar with a picture of my lab that was taken. I think, well, Divya is still in the lab in Sungwon, so it was taken, I think, about a year ago. And one of the things I like to do is point out that how international science is, and how it's really important that we all cooperate, and that in fact this country keep welcoming immigrants into its borders. I should point out, in fact, that these 3 individuals in my lab, they're US citizens, but each of them have parents that were basically came to this country from somewhere else. So thank you very much. And if there are questions, I guess there's a mechanism for answering them that's not your typical question. Sorry, this is just other slides. Thank you.
Thank you, Dr. Privis. There are questions, and we had about 200 people at least logging in. So I think your talk, as we expected, would be a lot of interest to people around here. So I'll start with the questions. The question from Wei Ni is, hi, to my understanding, the mutant P53 that regulates sterol biosynthesis is the loss of function P53. Then how about the gain of function P53? Many AML possess the non-mutational gain of function P53. Do you think these play a role in sterol regulation?
I think what you're asking is, to what extent, in looking...
Does the gain of function P53 have any, do you see the other side of what you showed, the gain of function P53 upregulating sterol? Right?
Well, we've never looked in AML, okay, to be honest. So I don't really know if mutant P53 plays a role in that or not. I know Scott Lowe has done some work in AML, and he has evidence for real gain of function of some mutants in those AML cancers that in fact have mutant P53. I can't really address its role in mevalonic pathway, unfortunately. And I think it's a really good question, to tell you the truth.
Okay, so Veronica Rando asks, how is the ferroptosis pathway altered in a P53 mutant setting? Would MDM2 inhibition also decrease cell death in this setting?
That's a fabulous question. And I wish I knew the answer. And it goes to something even more complicated, which I didn't want to touch on. And I sort of was trying to decide if I should include this. But in other settings that we've definitely confirmed and extended other people's observation that in certain cell lines that don't have P53, you need MDM2 and MDMX for survival. It's the exact opposite. So as soon as you initiate ferroptosis, there's actually a switch. And we don't know whether that switch takes place directly on MDM2 and MDMX or some other aspect of the cellular environment or both. But at least the data that we have suggests that MDM2 and MDMX can regulate cell survival when you don't have ferroptosis. In other words, you lose them, you have less, cells start to die and arrest. So it's the exact opposite, and it just depends on the context again. We've actually asked whether mutant P53 plays a role in this, and so far we do not see this. Now, does mutant P53 play a role in regulating ferroptosis? We don't know. But I should point out that one of the few natural ways you don't have P53 in cells, or you don't have wild-type P53 cells, is in cancer. So I actually don't know the answer to it. We've tried to do some experiments to address that, and so far we haven't found anything conclusive. I think it's a great question.
Right. So Jim DiCaprio asks, does the combined knockout of P53 plus MDMX eliminate the effect of MDMX inhibitors on ferroptosis?
Well, in a way, we're doing that just by knocking out P53 and then inhibiting MDMX. We haven't actually done, Jim, if you're asking, are we combining siRNA against MDM2 and P53? We haven't done that experiment.
Or I think the specificity of the inhibitors, right? So if you don't have MDMX and P53, are your inhibitors still doing something? Maybe another way of, but that's it.
Well, we actually, the other experiment we did, which was we actually overexpressed MDMX in cells and found that it caused a lot of extra ferroptosis. But when we used an MDMX mutant that couldn't interact with MDM2, it wasn't nearly as, let's say, cell death causing. You didn't get as much cell death. So that implies that, it wasn't a full rescue. So one thing we can conclude is that maybe you both need the complex, but MDMX alone may also have some role in this. And we don't know what that role would be.
Fair enough. Stephen Johnson asked a very interesting data. I was wondering if you've looked at PPAR alpha binding to chromatin. Protein levels are unchanged, but what about DNA binding?
We haven't looked at that. And we do have a co-IP with MDM2 and PPR alpha, which I didn't show. We haven't looked at that. That's a really good experiment to do. I think it's being done actually, but I don't know the answer. I have to say there's not fabulous antibodies against PPR alpha. And also a very weird thing was that we were never able to knock it down with siRNA. So I have no idea. I've never heard of that before, but we really couldn't.
So another question, which is, I guess, more philosophical, is from Max. I was wondering from an evolution Darwinian perspective, what would be the reason to select P53 involvement, I guess he wants to say, in so many distinct biological pathways?
You have another 2 hours? No, I mean, actually, I don't, even if you did, I don't. I mean, no, even if you did, I don't think I could spend more than say, I think it's a fabulous question. I don't know the answer. I mean, I think, you know,
all of us employed.
We're yes, exactly. I think, you know, the analogy of blind men patting an elephant is a really good one. I mean, we each have our own little corner of looking at it. I mean, I think, you know, I guess the one thing that I think keeps me motivated really in a way is the plight of Lee-Fermani patients, because these are human beings that have inherited a mutation in P53 and their cancer predisposition is through the roof. Interestingly, it's very specific kinds of cancer, but it's real, it's tragic. And I mean, it really does give us, I think, motivation to keep working on this gene in some way.
Question from Hua Lu from Tulane, I think.
Oh, Hua, how, this is really.
Missed you and your talk.
I thought it would just be people at Dana Farber.
Yeah, well, this is the beauty of Zoom. You could have people.
I guess so. Hello, Hua, how are you?
So he's asking you, does MDM2 or MDMX regulate ferroptosis in response to P53 activation? Is this cell specific? And also, did you look at the ferroptosis regulation by MDM2, MDMX in T cells or B cells?
No, we did not look into. It's not specific to the 2 cell lines we've worked in. We've looked at other sarcomas, and there's a very striking correlation between MDM2 amplification and sensitivity to ferroptosis. It's very striking. We've also looked in rat brain slices and seen something very interesting implicating MDM2 and MDMX. We have not looked in T cells or B cells, to my knowledge. I know that there is a program to look at B cell lymphomas in Brent's lab, and I don't know whether they're focusing on MDM2 or MDMX or not, but I can ask.
So the last question, I think even for time constraints, is from John Abrams, asking any evidence that long-term statins impacts incidence of P53-driven cancers?
I'm sorry,
Evidence that long-term statins, I guess.
Oh, yes. What is the role of statins in cancer?
Yeah, and,
that's a question I always get asked, and I've looked in Medline, in PubMed, and there are some of these, what you call, when you combine a whole bunch of clinical trials, meta-analyses or something like that. And so I found 15 meta-analyses of treatment of many different trials of statins. And here's what I found. 13 of the 15 showed a protective effect of statins in protection from liver cancer. I'll get to, and 2 said there's no effect. So I don't know what that means, but 13 out of 15 is pretty good. But liver cancer itself in humans is complicated because of the potential role of, for example, hepatitis viruses in it. And so we don't know where that would fit. But one thing I will say, which is so ephemeral, which is Chun Hao repeated the experiment where if you pre-treat the mice and then do his experiment, they get fewer tumors. But only once did he do an experiment where he started the tumor formation and then gave the mice atorvastatin, and that didn't seem to do anything. That's a one-off, and who knows whether that's a real result or not. In other words, whether they may be well protective against certain forms of liver cancer, it's not clear they could be used as a treatment. But that's, it's very, very speculative.
So I don't know whether we have time for one more, but there is one more, and I think Claudia will cut us off when we are done.
Well, don't do it.
It is, do you have evidence that mutant P53 also regulates SREBP1 and other aspects of lipid metabolism?
Great experiment. We've never, great answer, great question. We've never done that experiment. Never. So somebody should do it.
I think that's it. It's 105, and I was told 105 is our cutoff. So other questions, if they come in, will be sent to Carol, and I imagine she will if she can get back to you guys. But I want to thank Carol for giving this wonderful talk and taking the time and also the trouble of doing it on Zoom. Thank you very much. Thank you all for attending.
Thank you very much. It was really an interesting experience, and it's easier to do it by Zoom in a way.
Well, the travel.
Now I can go make a cup of tea.
All right.
Okay, thank you so much, Panjan. I really liked meeting you. Make contact with me if you want to discuss anything.
Absolutely. I will. Thank you so much.
Take care.